welcome. My name is Niklas. I got the honor and the pleasure to be your host for the next 45 minutes and guide you through this webinar. We have 45 exciting minutes ahead of us with this wonderful topic of the presence of non-human DNA in human DNA samples. So what we basically want to talk about is what else than human DNA do we find on the buccal swabs when we do our analysis. We have a very experienced speaker with us, so we'll introduce it to you and guide you through these slides. But first of all, before we get started, I'd like to thank everyone for joining in. I can see we already have over 200 people and 200 doctors joining in from all over the world. It's an honor to have you on board. It's our pleasure to have you on board, and glad to see that our community keeps growing. Now, before we get started, one thing I would like to point out and show everyone is we want to do this webinar as interactive as possible. So if you have any questions popping up, you will find on the lower part of your screen a Q&A button. And it looks exactly like the screen I'm showing right here. So if you click on this Q&A, it will open up a window which enables you to type in your question. If you type in your question and click send, these questions will pop up for us, and after the presentation, we will have dedicated time to answer all of these questions. In case there are questions which we did not manage to answer, we will upload the results on our website and also send them via email. But you can already ask the questions during the webinar per se. And also, please be reminded that we have CME points for this course. So if you want to receive the CME points, please stay during the whole duration. At the end, we will send a link into the chat and guide you through the process basically on how to receive these CME points. Now, let me get to the good part. So we will talk about today's speaker as well. We have Dr. Najim Amitiana on board today, one of my favorite webinar speakers. He's Senior Director Bioinformatics Research here at Arcensus. Najim also holds a PhD in human genetics with over 50 years of experience in molecular diagnostics of hereditary diseases and cancer. And he has pioneered the application of next generation sequencing in the clinical genetics department at the VU Medical Center in Amsterdam, where he grew up and studied. And he is also responsible for the discovery of a few different and novel gene disease associations by using cutting edge technologies in the laboratory as well as bioinformatically. And he is really excited about anything that's regarding genetics. So Najim, it's always a pleasure to have you on board. You're very welcome, and we're all looking very much forward for your presentation.
Thank you so much, Niklas, for the kind words. And I'm happy that so many people joined this webinar, and I'm happy to share also some of the findings that we did in at our company. So I will just dive into it and start sharing my presentation. Okay, I just want to have a quick confirmation that you see my first slide.
We see it perfectly.
Thank you so much. Okay. So today, I would like to discuss with you some of the findings that we did at our company. Well, since we are doing whole genome sequencing for samples in a diagnostic setting, when we started doing this analysis, we stumbled upon the presence of non-human DNA, and this is what I would like to discuss with you and explain to you what we all identified. So first of all, I would like to start off discussing very briefly about how we go about when receiving the sample up to the DNA sequencing and how the whole genome analysis workflow looks like in a clinical genetic setting. And then I will dive into the non-human DNA contaminants that we identify in these human samples, and then briefly touch upon how this impacts the genetic diagnosis. And then I would want to leave the floor open for the questions, which I will attempt to answer. So usually whole genome sequencing or diagnostics in general is performed on DNA from cells from individuals. And DNA can be obtained from different sources, and one of which is the oral sampling. And oral sampling is actually an excellent source for such an analysis because it's non-invasive, so you don't have to go with a needle into somebody's body, and it is very quick and easy to perform. It has been validated as such. And finally, it is also very easy for the logistics. You can just swap the sample yourself. It can be done by the individual themselves. And this can be just sent by regular post at room temperature. So there's no hassle regarding biological materials using sophisticated materials or any intervention via a medical expert. When it comes to the sampling using buccal swabs, I just want to focus here on buccal swabs instead of saliva, which is the other oral sampling approach. So the buccal swabs, usually there is this device that is packed in a sterile package, and there are instructions usually provided to the individual who needs to do the sampling. And as you can see, the important device looks like this, that has a brush at the end, which is important to keep sterile, so do not touch with your fingers. And then once you have this, you just unscrew this swab, and then you just strike the inside of the mouth, of the cheek, and then you do this at both sides. And after doing so, you just put the swab inverted in the tube that contains a solution that will preserve the samples that has been collected, and then you just mix briefly by turning this flask up and down for about 15 times, and then you put it in the mail, and that's in principle it. So when doing so, mainly what is obtained are the epithelial cells, which are at the inside of the cheek of the mouth. However, what can happen is that there might be also non-human cells that can be obtained, and this is what I'm going to be talking about. So just to give you an idea about how this looks like, if we just take the sample that has been taken from what I showed in the previous slide, and you will look into what is present under the microscope, and then this is an example from a buccal swab in the left picture. You see that there are different cells. Mainly the cells that you encounter are epithelial cells, or also known as squamous cells. And they can be either keratinized or non-keratinized. It's in principle not a problem, because eventually, since we do this for whole genome sequencing, it doesn't matter which type of cell we sequence as long as it's a human cell. And on the other side, we see an example of a sample that was obtained from saliva. And as you can see, besides the squamous cells, which you also can see in the left panel, you also can identify cells that look a little bit smaller, which are depicted in blue, and these are the leukocytes. These are the immune cells. In principle, it's all fine because both of them are derived from a human origin. And what is important to know is that although the cells are very big, the most important part of the cell that we are interested in are actually those blue shaped particles that we see there, because that's eventually the core of the cell that contains the DNA sample that we are interested in. And it's the same thing for the leukocytes that we see in the saliva section. However, for some samples, what you can see, besides these large cells with the core that contains the DNA in the middle, we often also identify, or there is also the presence of bacteria, which is depicted here in this very tiny rod-shaped dark particles that we see all over here. So this may be an example of an exaggerated sample that has a lot of bacterial in it. And nevertheless, it can happen that samples may contain also this kind of bacteria. And once this is also present, because the DNA, once it's isolated from a sample, it does not discriminate between a human cell or a bacterial cell, since the DNA more or less has the same format in both types of cells. So now I would like to briefly demonstrate how the workflow goes in a clinical setting. Once the sample is taken, then DNA is isolated from the sample, again, when the DNA is isolated, it doesn't discriminate between DNA from human cells or non-human cells. And then all the DNA is then subjected to whole genome sequencing. And there is no enrichment involved, meaning that the DNA is sequenced as a whole. There is no enrichment for human cells, nor specific regions of the human cells. And then once this data is provided, it will be analyzed using different tools, which I will discuss briefly. And then once the human genome is sequenced, then the relevant variant is identified, a report is then sent to about the subject to the physician or directly to the individual who's interested in the report. This part, which involves the analysis, in principle, the sequencer provides what is called the raw reads, and this is done in the format of FASTQ files. So this contains all the tiny bits of the genome, regardless of the source, which is sequenced. At this stage, only reads are obtained, and then an alignment takes place where we try to actually rebuild the genome from these tiny bits of sequences, just to identify where exactly in the genome they fit. And once this process is finished, then the variant detection starts to take place, because at this stage, we have reconstructed the genome from a sample. And then at this stage, where we try to identify the variants, we try to look for differences between the sample that is sequenced and a reference genome. And this reference genome is a fixed reference that contains the DNA sequence that is used universally by all diagnostic labs, and then the variants are identified. And once the variant is identified, then annotation is performed where we try to provide context to the variant. So what does this variant mean? On which chromosome is it present, and what kind of effect does this particular nucleotide change has, for instance, on the protein, if it is affecting a protein, and whether this particular variant has been identified in any of the databases that are available, and whether it is potentially harmful, whether it is associated with disease, and so on and so forth. At this stage, there are about 150 databases that are used to provide additional context to this particular variant. And once all this information is provided, then the filtering and the prioritization to identify the candidate or the relevant variant for reporting. Just to put this in context, at this stage, when the variant has been identified, we end up, if doing whole genome sequencing, we end up with about 5 million variants. And eventually, what is reported is usually a handful, if maybe in most of the cases, even a single variant. So we have to do the filtering to go from 5 million variants to a single variant that needs to be reported. So this is the normal workflow in the diagnostic setting. So just to give you an idea how this would look like, when you do whole genome sequencing data, you end up with roughly starting from about, let's say, 500 million reads. So you obtain 500 million reads for a single sample. And you can see the more reads you have, which is depicted on the x-axis, the higher the depth of the coverage. And the higher the depth of the coverage, the more confident you are that a specific variant is true, and you don't want to have many variants that are artifacts, and therefore, to reduce the number of artifacts, you need a higher depth of coverage. So you can see the more reads, the more data you obtain per sample, the higher the depth of coverage. As you can see, there is a nice correlation between the number of reads obtained per sample and the average depth. However, this correlation is even more robust when we only focus on the reads that are mainly coming from the human sample. And as you can see, the correlation coefficient when only focusing on the reads that are actually aligning to the human genome reference, so reads that are actually only coming from the human DNA, the correlation coefficient is almost excellent when only focusing on reads aligning to human. And Whereas if we also get all the reads that are obtained from a sample, we see that the correlation, although very good, it is less than when looking into the exclusive human aligned reads. And this is mainly because many of the reads do not align to the human reference genome. And this suggested to us that there has to be different types of DNA present, which is not coming from the human DNA. So to be able to identify then what are these reads that are non-human, to do so, we took the raw data or the bound files, so the data that has been aligned to the human reference genome. And once this bound file is created, each read that is aligned, it will receive a flag. If the DNA is not aligning to the human reference genome, it will get a flag that it's not aligning to the human reference genome. What we did, we took all those reads that didn't align to the human reference genome, and then we realigned using a tool that is called Kraken, which in principle takes all the known microbial genomes that are available and published, and we align to all those different genomes, and then we try to identify the percentage, the composition, and the number of reads to each of those different microbial genomes. And then we can classify them on taxonomic domains and try to identify which microbial families and species have been identified. And just to show you and bring this in perspective, we took the genomes that have been published in the NCBI database, and we used these 4 different domains, which are genomes coming from bacteria, archaea, viruses, as well as eukaryota. And once we delve and go deeper or get more granular all down to the species, we ended up with about 17,000 different species that we have looked at. So once we have aligned the data to the human reference genome, then we can start to identify and look into the samples as to how many reads or what's the percentage of the reads per sample that did not align to the human reference genome. And what you see here is that if we set the cutoff of, let's say, 5% of the reads that did not align to the human reference genome, then we can see that when it comes to the buccal swabs, and this is this gray bar over here, we see that more or about 82% of the samples have more than 5 reads that didn't align to the human reference genome. We don't know yet at this stage to which genome these reads then to align, but this is something that we can explore in the next step. This just goes to show, if we focus on the 5% non-human aligned reads, what this NHMR stands for, then more than 80% of the samples contain those types of reads. And for the saliva, also around 80%. But if we say, let's set the cutoff at 1% of those reads that do not align to the human reference genome, then the percentage rises a bit higher, which sounds logical. So this shows that almost all samples demonstrate the presence of non-human DNA, especially when taken from buccal swabs and saliva. Now, if we look, for instance, at the percentage of buccal swabs and the percentage of non-human mapped reads from the buccal swabs and the saliva, then we see that there are some differences within the different groups within the buccal swabs or the saliva. Some samples have hardly any non-human mapped reads, whereas others have a lot of non-human mapped reads. And the same thing holds true for the saliva. But in general, if we look at the 2 different ways of collecting samples, there doesn't seem to be a difference between whether the sample is extracted or is collected from buccal swab or saliva. They both show this large deviation, but in general, they have more or less the same average non-human mapped reads within both types of samples. So what we have investigated the following. We asked ourselves which parameters actually influence this contamination. So we looked at the gender of these individuals, we looked at the age and the geographical region from where the samples came from. And also we looked a little bit into what type of contaminants are present in the samples. And finally, we wanted to know whether the level of contamination would have any effect on the genetic diagnosis, which is, in my opinion, one of the most important aspects when it comes to whole genome sequencing in the context of genetic diagnostics. And just to put this in perspective, because I realized I didn't show you the numbers, we analyzed over 650 samples, and these samples when combined the buccal swabs and the saliva. So if we look at the gender to see whether there is any difference between males and females when it comes to the level of contamination or the level of non-human mapped reads, then there is hardly any difference. So there is no difference that could be explained by the gender. They have an equal difference when it comes, or equal range of non-human mapped reads between the different genders. And then if we look at the age, we try to do this as granular as possible. So you see on the x-axis are the age groups. So the first bar shows the age group between 0 to 1 year, and then from 2 to 3 years, from 4 to 5 years, and so on and so forth, until we set the last group from 20 years and older. And as you can see, within each group, there's still some deviation when it comes to the level of non-human mapped reads. But what we can see is that there is a trend that all the way when we have infants of the youngest age group, they have the lowest contamination in terms of the average. And when the children get older, they start to pick up on the non-human mapped reads, which is mainly composed of bacterial DNA, which we'll see in a minute. And then it seems to also gradually decrease once the individuals get older. However, we have to admit these numbers are a little bit on the low side, and therefore we cannot make any firm conclusions at this level, but there seems to be a trend. So then we checked whether, just based on the region, so from which region we have sampled the cases, to see whether we can find a difference in the percentage of these non-human mapped reads. And we clustered the samples which came from either Pakistan, Albania, Germany, and the Middle East. And on top of each bar, you can see more or less the number of samples that belong to this particular group. And although there are some slight differences between the regions, however, the numbers within each group deviate slightly, or deviate when it's drastically between some of the groups. And therefore, similarly, there seems to be a trend that, for instance, the cases that came directly from Germany, they seem to have the lowest non-human mapped read. However, the numbers, again, are too low to be conclusive when it comes to claiming whether a region would affect the percentage of non-human mapped reads. And then after this endeavor, we wanted to know whether what type of different bacterial families are present in this samples. So in this case, we looked at the level of the families, because within each family you can go into different species, and then the picture will become way too complex. And therefore, we just showed in this particular graph, you can see the number or the percentage of the different bacterial families that's present in all the samples. And as you can see, the Streptococcaceae bacterial family is the most prevalent, which is present in about 90% of all the individuals. And then it's followed by 44% carried by all the individuals by this Neisseria bacteria. And then finally, there are more, but also on the third place, there is this Pasteurellacea bacteria. You already see that I'm not really a microbiologist, but I try to do my best. And just to give you an idea, this data is only when focusing on reads that are at least 5% non-human mapped reads. Because if we would extend this to include lower percentages, then although the picture will still remain the same, the numbers would increase, because we would allow also lower amounts of non-human mapped reads to be included in this graph. And this is why we set this at 5%. And then if we look at the bacterial, at the level of the bacterial species, not surprisingly, because the Streptococci were the most prevalent family, this is also the most prevalent species that was identified. And to be more specific, the Streptococcus mitis seems to be the most prevalent, which is in more than 50% of the cases of the samples. And altogether, these different species, are, as a matter of fact, part of the oral flora. So this is not really exceptional. And one can imagine that in principle, this is not, this level of presence is not really clinically that much relevant. However, it becomes relevant when the amount of bacteria gets the chance to overgrow, and it may then cause any other many issues such as caries and dental problems. And then we wanted to know, because we had from the Pakistani cohort was the largest cohort, consisting of more than 500 individuals. And since we did have the information from the regions of those different samples, we tried to identify whether there is any difference in the type of bacterial families that has been identified. And as you can see in this graph, as a matter of fact, there is no clear distinction. The region, the sub-regional location of the sample does not affect the type of bacterial families that has been identified. As you can see, the Streptococci is the most prevalent, but it's the most prevalent also in all different regions from Pakistan. And the same holds true for all the other samples. There might be some exceptions here and there. However, it is not clear whether this is significant, and we want to investigate this a little bit in more detail to see whether indeed this could be explained by some factors that are regional. However, to be able to do so, we would need to have some more extensive data from the individuals. And just to give you an example, if we would focus on a single sample, and I don't expect you to all read and read all these different letters that you see here, but this is just an example of, if you dig into a single sample, how the distribution of the different bacterial species would look like. So for instance, once we start doing the analysis, you can identify that the majority of reads in this particular sample belong to bacteria, and then you start to divide you start to branch out based on the type, on the phylum of the bacteria, the class, the order, the family, the genus, all the way down to the species. And then you can specify and identify very specifically which type of bacteria is present. And as I mentioned already at the beginning, we did not only look into the bacterial genomes, we also interrogated the archaea genome as well as the viruses. And indeed, viral genomes were also identified, albeit at a little bit at lower percentages when it comes to these non-human mapped reads. And also what I want to mention here is that the majority of the viral DNA that has been identified were usually coming from the viral species that are actually have the bacteria as the host. So usually, whenever we have samples that have higher levels of bacterial contamination, these are usually the samples that contain also the viral species, which usually are inhabitants of this bacteria. So then finally, and I think this is the most important when it comes to the clinical diagnosis, we wanted to know whether there is an effect of the level of non-human mapped reads, or the level of contamination of the human sample by this bacterial species on the diagnostic yield. To do so, we initially categorized all the samples that we have within the buccal swab or the saliva into 3 categories. We categorized them as high, when containing more than 20% of the non-human mapped reads, as medium when they contain between 5 to 20% of the non-human mapped reads, or as low whenever they have less than 5% of the human mapped reads. And then we only focused on the samples that had either a positive or a negative diagnostic result. So we ignored a couple of samples. This was the minority for which not a genetic diagnosis was requested for a specific disorder. For instance, if it's a carrier testing, this were ignored. We only focused on clinical cases that had symptoms, and we wanted to know whether we can identify the relevant variant that could explain the symptoms, or we could not identify this. So then we looked into this, and what we could identify, we saw that, as a matter of fact, there is no difference between the sample, the level of contamination, and whether a sample would be diagnosed, yes or no. As you can see, in all 3 categories, so in the high category, we were able to diagnose 74% of the cases, and about 78% in the medium case, in the medium group, and a similar percentage as in the high was also identified in the low, which was a little bit surprising, because we were expecting that the more bacterial contamination you would have, the worse the diagnostic yield would be, as this bacterial contamination would somehow reduce the coverage, so the depth of coverage for the samples, and therefore would make the identification of these relevant variants difficult, which was not observed. And with this, I would like to conclude. As I could demonstrate, I hope that you saw that the usage of oral sampling to obtain biological material for genetic testing is an easy and safe method. It can be applied and used by everybody who's able, who has the possibility to do so. And it is a suitable method for genetic testing, and as demonstrated, this with the high diagnostic yield of over 70%, which is not related necessarily to the buccal swabs in the saliva, but it's most likely related to the cohort that was analyzed, and it is suitable for whole genome sequencing and does not affect the potential non-human mapped reads contamination. And the presence of non-human cell contaminant does not affect the diagnosis. And with that, I would like to leave this, because I think this is one of the most important messages. And I would be happy to take your questions. Thank you.
Perfect. Thank you very much, Dr. Najib Mamtiani. You get digital applause from me, and I'm sure from everybody else as well.
Thank you.
I'm happy to see we have a lot of questions. We have also over 300 participants, attendees from all over the world. And I'd be happy to welcome Ahmed to support us in steering the conversation and to go through all the open questions and make sure we answer all of them.
Okay, so hello everybody. We see a lot of questions. We'll try to go through them, and those who we don't manage, we will reply them digitally. So I'm combining a few of the questions for you, Najib. There are a few questions from Dr. Gilbert, Dr. Jamshed, and a few others as well. They're asking about the chances of contamination and also regarding the special requirements before sampling. So any special requirements before sampling?
So one of the most important requirements is that one should not eat just before sampling. It is usually recommended to sample at least 30 minutes after one has had lunch or dinner, just to make sure that you don't get other contaminants besides bacteria, just other contaminants that may affect actually the extraction of the DNA. So you want to remove all known cells and whatever is present in the food that is being consumed. there was another part of the question, probably I didn't get that. Can you repeat that?
Yes. What are the chances that there could be a contamination? So the percentage of chance.
So as you could see, the percentage of having contamination, so I would say there is always contamination. The problem is usually the level of contamination, because there is always a microbial flora in the oral cavity. So one cannot escape this. And this is, again, not an issue whatsoever, because, as mentioned earlier, the level of contamination does not affect the diagnosis. And therefore, the level of contamination is also depending on many factors. So the oral hygiene, and I guess if one takes care of the oral hygiene, then the contamination will be less, and vice versa. If one doesn't brush the teeth, for instance, that often, then obviously people who like to consume a lot of sugary food would also increase the level of bacterial contamination. So it's a little bit difficult to answer this question. It's really depending on the diet and the lifestyle of the individual.
Thanks. There is a phrase in the questions. It's glycan mucin synthesis is non-human DNA. Is this expression correct in your opinion?
Can you repeat that?
It's glycan mucin synthesis. Is it the same like non-human DNA, glycan mucin synthesis?
Well, this sentence describes a process. It doesn't describe an entity. You know what I mean?
Okay.
If I say glycan, usually these are not. These are enzymes. These are proteins, and in principle, this is not affecting the extraction of the DNA, because when we extract the DNA, we are mainly interested in cells that have a cell core, the nucleus that contains the DNA.
Perfect, thanks. And there is a question from Dr. Kriati. She's asking, which type of sequencing do you perform with this sort of sampling? So which sequencing do we use in Arcensus?
Yes, so we do whole genome sequencing using the Illumina machine, and to be more specific, we sequence using the protocol that is pad end and has a size of 150 base pair from each end.
Perfect. There is another question about the age group. So you presented between 4 to 5 age group, there was level of contamination was higher. Any comments on this?
I think this is the group usually that eats a lot of candy, and it's usually the group that's usually difficult to convince to take care of the oral hygiene. To me, this is speculation from my side, because to be able to provide a more accurate answer, it would be important to do questionnaires, mainly of the parents, to be more accurate. But this is my speculation. I think it's because they consume a lot of candy and they do not brush the teeth that often.
That's correct. There is a really interesting question. Is there a cutoff below which you would say the sample failed, or above which it's passed for sequencing or analysis?
I think this is a very interesting and important question. However, as we have shown from the data, there is no specific cutoff. As a matter of fact, we have some samples that have about 70% contamination by bacteria, and nevertheless, we were able to identify the disease-causing variant in the sample. So mainly, it's not actually the percentage of contaminant that is present that affects the diagnosis. It's more related to whether a sample is genetically affected, because this you don't know beforehand when you start doing the analysis, unless the cohort is really well defined and it should be clearly genetic. And the other part that could affect the diagnosis and therefore a set-off needs to be set is whether the variant is present in a specific region that is either difficult to sequence or it's easy to sequence. So in principle, there is no cutoff, but what we usually do, we analyze the case regardless of the percentage of non-human mapped reads. And if it's negative, then we would request, and there is too much bacterial contamination, for instance, then we would request resampling using a different source, especially if the contamination is too high and it is not possible to reduce this contamination, because we could also ask to do the sampling in a more careful way to avoid such contaminations. But in short, there is no specific cutoff.
Perfect. Thank you. So there is a question from Dr. Munir. He's asking, can this be served as an evidence for evolutionary relationship between the other species, or is it just because of some contamination?
That is a very broad question. I'm not sure where we should go with this, because in principle, what I showed, all these bacterial families and species that have been identified, these are already known species that are inhabitants of the oral flora. So there is nothing new. And to be able to look into these kinds of evolutionary projects, it would mean that we actually have to investigate into and identify the species, but also investigate at the nucleotide level. So try to identify specific mutations that, for instance, point towards adaptation to specific environments or specific diets. And this was not exactly the scope of the study, but it would be interesting to follow up, but it would require maybe a PhD student or two to investigate this.
Right, makes sense. So we are short of time, maybe just a few more questions. The rest we will answer online. So mouth infections, there's a question about mouth infections. Does it affect if somebody has a mouth infection and we sample it? Is it more contamination or is it the same?
That's a good question. To be honest, as far as I remember, we didn't have a specific sample that would belong in such a category. But what I can imagine that depending on the infection, obviously, but usually an infection, depending on also the stage at which the sample is taken, an infection is usually also accompanied by inflammation. And if there is high inflammation, for instance, one would suspect the presence of high levels of leukocytes, which would again also provide a higher percentage of leukocytes, which are in principle human cells. But I cannot really address this question specifically, but just on the surface, I would assume, yes, if there's an infection, the level of contamination would be higher. And therefore, it would be nice indeed to follow up on this, for instance, to assess whether the individual at the moment of sample collection, whether there was an infection present, yes or no. Unfortunately, this information was not made available to us. Otherwise, this would have been looked into as well.
Perfect. And maybe the last one, it's a very general question. How long does it take for the analysis for the whole genome? So the turnaround time for the analysis.
So here at Arcensus, we use a turnaround time of 20 working days. So in principle, the entire workflow is depending on separate processes. The sample collection, the providing the sample, and the arrival of the sample at Arcensus, and then performing the sequencing and then the data analysis. So if there are no issues among these different processes, then a sample analysis can be executed definitely within these 20 days. If the sample took too long to arrive to us, or due to whatever reason, then, or there was a machine failure, obviously, this could take a little bit longer. Nevertheless, there are always redundancies built in the process. If at one step something can go wrong, then there are alternatives that can be applied so to speed up the specific step and to have the results as mentioned earlier within these 20 working days.
Thank you both very much, Najim, Ahmed. It's as always super exciting listening to these conversations and to the answers. Thanks a lot. Now it's up to me to wrap up for today's webinar. As stated, those questions which we did not have time for answer, we will make sure to document them and send them afterwards. Now, the three last points I would like to share with you is, on the one side, if you enjoyed it today, of course, you're more than invited and more than welcome to join our next two webinar series, which are coming up. It's going to be on the July the 27th. Our founder and CEO, Professor Dr. Arndt Rolfs, who is having the fifth part of our webinar series, Unexpected Diagnosis Based on Whole Genome Sequencing, where we'll use real-life cases and show cases where the disease was not discovered using whole exome sequencing and how we changed that using whole genome sequencing. And then we have on the 8th of August, Dr. Wallace Krajina. She's Senior Director Clinical Programs, and she will talk about genetic disease prevention with clinical actionable incidental findings. And sorry for the date, it's the 10th of August. You will also receive this date in the email afterwards. So the 10th of August and the 27th of July. Now, that was the first thing. Now, the second thing I would like to say before we finalize, it's we talked about the attending and CME certificate at the beginning. If you would like to receive such a certificate and the associated points, please make sure to fill out the form, which will now be posted in the chat. You will find a link, and this link will guide you to a digital form. Please fill out this form correctly. And once this is done and has been issued to us, we will provide the certificate within 7 working days. If you should have any questions in the meantime, reach out to us, visit our website, and contact us, and we will make sure to help you. And that's it basically from our side. So the last point is saying thank you for everyone for turning in. It's a pleasure having everybody on board. It's an honor to see how many people from all over the world take time to listen to these webinars. And we are very much looking forward to see you in the next ones again. Thank you very much, and stay healthy.